and welcome to yet another insightful episode of Beyond Shodh. I am your host Dr. Sonia Balyan, and today we have the privilege of introducing a distinguished scientist and an educator, Dr. Deepti Jain, who is an associate professor at the Regional Center for Biotechnology, Faridabad. She has made significant contributions in the field of structural biology, particularly in understanding the molecular mechanism of regulation of gene expression in pathogenic bacteria such as Pseudomonas aeruginosa. Her research involves utilizing an integrated approach, combining the structural tools, biophysical techniques, and biochemical methods and functional in vivo assays. This led to the insights into the regulation of flagellar, biofilm, and antibiotic-resistant gene expression. Dr. Jain has leveraged these mechanistic insights to develop novel targets for the development of anti-biofilm agents. Throughout her career, Dr. Jain has been recognized with numerous awards and accolades, including the Early Career Award from SERB and Innovative Young Biotechnologist Award from DBT and the Jane Coffin Child Memorial Fellowship for Medical Research in USA. Her passion for research and teaching has inspired countless students and fellow researchers. In this episode, we'll delve into her remarkable journey, her impactful work, and her dedication to advancing the knowledge in biotechnology. Join us as we uncover the fascinating world of science with Dr. Deepti Jain. Welcome, ma'am, to Beyond Shodh, and thank you so much for providing us the opportunity to feature your research and your journey.
Thank you, Sonia. That was quite an introduction, and I'm quite thrilled to be on this episode, and I look forward to interaction with you.
Thank you so much. Ma'am, to start with this, can you please share with our audience a little bit about your research and your professional journey?
Yeah. So, I did my bachelor's from Delhi University in microbiology honors, after which I moved to IIT Roorkee for MSc in biotechnology. I joined National Institute of Immunology in New Delhi for my PhD, and it was during the PhD coursework we had to decide the area of research that we wanted to pursue, and I realized that the best fit for me would be structural biology. So I joined a structural biology lab and continued my PhD, and after completing PhD I moved to Rockefeller University, where again I pursued structural biology as the primary technique that we were using to address mechanisms in transcription regulation. After that, after completing my postdoctoral studies, I came to NCBS as a visiting scientist and spent few years there before I joined RCB as an assistant professor in 2013. So it's been 10 years now. I will complete 10 years next month, and yeah, this is a good milestone for me and my lab.
Sure, ma'am. So, ma'am, what exactly are the areas in which your lab is working currently?
Yeah, so we address transcription regulation in bacteria. Particularly, we are interested in pathogenic bacteria such as Pseudomonas aeruginosa. We also work with Staphylococcus aureus. Both these organisms are pathogenic. They are also have been listed in the critical category pathogens in a list defined by WHO as well as by DBT. And what we are interested in looking at are the flagellar genes in Pseudomonas, how they are regulated. So flagella is a nano machine present in the bacteria which helps the bacteria to propel forward. So it is a locomotory organ. But besides being helpful for motility, it is also important for pathogenesis, it is also important for virulence, and it is also important for its ability to cause biofilms. So biofilm forming bacteria, they encapsulate themselves in a polysaccharide matrix, and that makes them impervious to antibiotics, so leading to antimicrobial resistance. So both Staph aureus as well as Pseudomonas are known to be notorious for causing biofilm related infections. So we look at how the flagellar genes are regulated in Pseudomonas aeruginosa, and also the pathways which are important for antimicrobial resistance in Staphylococcus aureus. So the primary technique that we use in our lab is X-ray crystallography. It's a technique of structural biology, which is what I gained experience in during my PhD as well as in my postdoctoral training. And because I had a background in microbiology, so this
All
was the
gel
best
together.
thing to combine these two expertise when I started my lab. So X-ray crystallography gives you, it's a unique technique which actually gives you atomic level details of the molecular interactions that are taking place in the living system. So basically we are looking at chemistry of interactions that is happening in the living system, be it plant, be it bacteria, be it humans. This technique can be used for looking at these atomic level details in any system. So we look at these, how molecules interact with each other, what is the chemistry that is going on, and we are using a technique of physics to address the questions in biology. So it's truly an interdisciplinary technique that helps us to look at mechanisms in detail. Yeah.
So ma'am, through your research, what leads are there on which can be translated?
I think over the years now we have generated a wealth of information which is in structural information coming from the transcription factors which are regulating both flagellar as well as biofilms in these bacteria. So we have a lot of structures of transcription factors now and in their functional states. So these structures can now be targeted for drug discovery. So we also now have begun, because now we have accumulated a lot of structural data in our lab, and we have now begun looking at how these structures can be utilized as targets for drug discovery application. Particular emphasis is on trying to find something for biofilm related infections. So biofilm related infections are very, very difficult to treat. They are usually formed in patients who are suffering from who are immunocompromised, suffering from, say, for example, cystic fibrosis or burn and wound in patients. They get infection from Pseudomonas aeruginosa, and these are biofilm-related infections. And as I said earlier, that the antibiotics do not work in such scenarios because the organism covers itself with a exopolysaccharide matrix, making it very difficult for the antibiotics to penetrate. So currently only surgical removal is the answer, and very aggressive concentrations of antibiotic are given to the patients. So we're trying to see if there is a possibility of breaking these biofilms so that antibiotics can reach the site of infection. And we are hoping to utilize all the structural information that is generated in the lab to do structure assisted drug discovery.
So ma'am, this biofilm formation, is it the natural process or it is an adaptive response of that micro?
So more and more research shows that it is actually the natural process. The organism prefers to go into the biofilm mode of life. And so there are two distinct modes of life in Pseudomonas. The flagellar mode of life, where the organism has a unique only single flagella, and it will swim around using the flagella. But when it enters into the switches to the biofilm mode of life, it loses this flagella. It becomes immotile. It adheres to surfaces, and it makes colonies of bacteria and covers itself with the matrix. And also metabolically, it's not very active. So therefore, treatment with antibiotics again becomes very, very challenging. So these two, how this organism switches between these two modes of life is also one of the questions that we are addressing in the lab. At the molecular level, what are the events that take place and what is the trigger that allows the bacteria to switch from one life form to the other is also under investigation. Yes.
So ma'am, if not by antibiotics, how can patients or doctor get to know that now this infection
Yes, so that's an important question because so diagnostics in biofilms is not very advanced, or it's not available. So there are very few things that we can do to check whether, but already the infection has spread at that stage is when people look at expression of alginate. So for example, Pseudomonas which are isolated from lungs of patients, they are rich in alginate. So the content of the matrix, it varies, and that has been used probably to diagnose this kind of infection. But yes, diagnostic is also something that needs to be developed to look at biofilm related infections.
Because when doctor keeps on prescribing higher doses
Yes.
of antibiotics,
Yes.
if that is not helping,
Yes.
so what is the next step? If
I
this
think
is
a
not-
lot of people
Yeah.
now are trying
Hmm.
patch therapy to be
Okay.
one of the that has shown some promises
Hmm.
to treat biofilm related infections, but still I think there's a long way to go. And newer antibiotics need to be discovered. Current antibiotics need to be modified,
Yes.
and looking at whether you can repurpose some of the available drugs. All of this needs to be tried.
And that this capacity of forming biofilm, is it a matlab, a canonical capacity of every microorganism, or it is
Some
specific?
microbes are known to form better biofilms, and Pseudomonas is actually used as a model system to understand how biofilms are formed. So it is a lot of a problem in Pseudomonas related infections, but there are many other bacteria that form biofilms, including Staph aureus, including E. coli, and so on and so forth. But in Pseudomonas, this is a huge problem, and it's in fact used as a model to understand how biofilms are formed in bacteria.
Very interesting work, ma'am. So how you got the motivation to pursue these areas?
Yeah, so I would give credit to my teacher in masters, Professor Ritu Bartwal, who taught me biophysics. And during her classes, I realized that this is a fascinating area of research. And in fact, I ended up doing my master's project in her lab, where I got exposed to reading NMR spectra. So we were looking at NMR of short nucleotides and even bound to drug molecules, some oligonucleotides bound to drug molecules. And I realized that this is a fascinating area. And similarly, when I joined PhD in NII, Professor Dinkar Salunki taught us the coursework, and after we complete the coursework, we're supposed to select the lab. And I realized that structural biology, because of this background in biophysics, would be the appropriate field for me. And till date, I'm quite excited about this technique. It gives me goosebumps when my students solve a new structure, because it's completely new, and it is our discovery. It's our we have solved the structure of a protein which nobody knew what it looked like earlier. Gives me a lot of excitement.
Because everything converges to protein ultimately.
Yes, eventually.
Every
Protein are the
regulation-
workhouse of the cell, and all functions, most of the functions are carried out by proteins. And we need to understand it to get functional insights. It's very, very important to look at structures. I see sometimes students are hesitant to take up this area of research, to pursue this, because they do not have a background in biophysics or structural biology. But now I have students in my lab from all different areas, like including microbiology, zoology, botany, medical sciences, who are feeling comfortable pursuing structural biology. I was lucky because I had this biophysics. So I think there should be a course in masters in structural biology, just to introduce to the students, and therefore they can be more comfortable pursuing this as an area of research during their PhD. So in fact, RCB MSc program offers structural biology course in the master's curriculum. So I think that should be included in.
Yes. Great, ma'am. So, ma'am, what is the most rewarding thing about research in academia for you?
I would say the interaction with the students when they make some even small discoveries, or when they come and show me with all their excitement, even as small a thing as a gel which has run very beautifully, and so it is very satisfying to see that excitement on their face. Also, very satisfying to see their journey, that when they join the lab, how naive they are, and they're a little lost, and how they evolved into scientists. That is very, very rewarding.
Coming to, you are a woman scientist and leading RCB in your area. Gender diversity in STEM is an important issue that we all know. And as a female scientist, you have, do you faced any unique challenge or the opportunity, maybe that creates some hurdle to pursue your path?
So I don't know of any challenge particularly for this reason. There were many challenges, but not because
of
the gender issues. But yes, gender balance in science is very important, very essential. We see that there are fewer women as the ranks are, yes, at faculty level or at, director level or at higher positions in the institute. And that needs to be addressed and that needs to be taken cognizance of. We need to accept that and take actions so as to, if that can be improved, the ratios can be improved. We see it because in my experience in last years, there are many female students who join PhD. In fact, sometimes we have more number of females joining PhD as compared to the males. But as we go higher up in the ranks, we see fewer and fewer women in these roles. So definitely we are losing these women to something, and that needs to be addressed, and that needs to, because it's very important for inclusion and diversity that we have women in different positions.
So ma'am, how do you balance your work, your research along with your family responsibilities?
Yes, so I think all women require a lot of support system, and that is very important. My kids have grown up in daycares, so that support system was very important for me, especially at the time when I was in NCBS. My kids were very young, and they have a very beautiful daycare on campus. So I can't imagine coming back to the lab into the sciences if not for that daycare. So there are small things like that. I think they go a long way in having women back into the labs, into research environment. So support is very, very important in terms of family support. My parents were very supportive of me pursuing a career in science, and they continue to do so. So sometimes when I travel, I request them to come home and stay with the kids. So that kind of family support and flexibility in working hours would go a long way in supporting women into sciences. So I think every institution, at the individual level, institution level, organization level, we need to have this kind of support system for them and a little bit of flexibility. We'll definitely see more women coming out of the science. I think my advice to women would be that they need, if the passion is there for science, they need to continue. There may be breaks, there may be obstacles, there may be career breaks, few here and there, but they need to come back. There are a lot of opportunities now. Some government schemes are there now, which they should make use of and try to come back to the mainstream. So it can get delayed, but it cannot, not happen. So I think.
So ma'am, in your lab, what type of ecosystem you prefer if you have women students?
I don't think there is a separate rule or any separate requirement for women. I have no less expectation from my women scientists than I have from the male. So I'm equally, I feel that they need to achieve as much as a male scientist in the lab. But the environment that we consciously create is friendly. They need to collaborate with each other. They need to be able to freely discuss with each other. There has to be, also I make sure that they are, they feel free to approach me in case there's a problem. The environment has to be safe for them. The environment has to be inclusive for them. And that goes for all the students, irrespective of whether they are males or females. So yeah, I don't expect anything less from them.
No, I don't mean less. I mean, sometimes, if women are married, they have to juggle between them. So little bit relaxation in time maybe can.
Yes. So that I said earlier, I think all they need is a little bit of flexibility and a little bit of support.
Otherwise, I think they are performing better than
absolutely.
You agree with this. So ma'am, could you please share any memorable experience or that could act as a turning point of your career and shaped the lead for your lab? Any particular discovery?
I would say I can narrate one incident, but I think that turning point maybe probably is yet to come. But challenges that we face, one of the examples that I could give you is that very early on in my when I was setting up my lab, one of my first students when he joined, and we were working very hard to solve the structure of the protein. And as we were collecting the X-ray data, we figured that the paper has appeared on the same protein, from the same organism by another group. And we both were devastated, and I was very young as a mentor at that time. I had to sort of hide my emotions, and already the student was in such a state. But so I think what got us through was that we both sat together, thought through this, whether we need to change tracks, whether we need to continue to pursue the same project. And I think after a lot of discussions and deliberations, we decided not to quit. And we continued pursuing, and we published several papers. And now I'm less concerned when I see a paper in the on the same protein in a journal. That time probably I was very young, and I was also concerned about how the student's career would shape up. But he's doing fantastically well. We published several papers on the same project, same structure, same protein. And so that made me more experienced. And this is, I think, one of the things which I learned is to not quit. When you're working on a project which is very, relevant, you will have competition. And one has to accept that, and you will find your way, and is what I think. And so now I don't get as disturbed.
Like these similar instances, I think happen in most of with
Correct.
most of us.
Correct.
Every time the work in which you are doing research, it's possible that somebody
Yes, if it's
also
interesting, there'll be more than one group which is interested.
He is working on his line, we are working on, but
Correct.
Again, when we publish, it impacts.
Yes.
It impacted the quality of journal, or they say that this is already published.
Correct. But it also is the story that you tell. From the same structure, you can analyze it in a different aspect of it. So I think we told a different story. We ultimately published it, and it was quite satisfying to see that we did not give up on the structure. We pursued it, and also it was thanks to that student who persevered and did not give up on it. Yeah, that is one of the memorable experiences.
Yes, ma'am. And I think now you have several other such experience with now, but you know how to handle now.
That's what I said. At that time, I was quite academically, I was young, and he was one of my first students. So I was disturbed, but I think we made it happen eventually.
What is your advice to early career researchers who want to make significant impact in biotechnology?
I think one of the very important thing is that they need to stay curious, and curiosity is very sort of a powerful thing, because it will drive you, and it is very, very essential to continue to ask why this is like this, how does this work, what is the mechanism. So when these questions will come to your mind, the students need to remain curious, they need to continue to ask, they will do very well. I think that is my only advice to the students, that don't lose that part of you, don't lose curiosity. That takes you a long way.
Yes. Ma'am, how do you see your journey, and how your upbringing and your family values shaped you as a leading scientist in your domain?
My father is in sciences. He's an organic chemist, and my mother has done double masters in Hindi and Sanskrit. They were very keen from the time we were very young that both the daughters and the sons have to have a career, and they worked very hard to support us. And and my father was very keen that we should pursue sciences. So from that time onwards, the interest grew, and after that I did not know what else to do, because I was so fascinated, passionate about pursuing sciences. So their contribution was to initially shape this, and also one thing we all learned, my siblings and me, was to work hard, and there's no substitute for that. And we continue to do that. So yeah, there's a tremendous amount of motivation and support that comes from my parents.
Now your kids understand that?
kids have become more independent because I'm less available at home, and so I think that's also in one way good. My mother also worked throughout. We were at home all by ourselves, and I think that allows you to become more independent much faster and take up responsibilities. So, and there is a positive to that as well.
Yes, ma'am. So, ma'am, what is your future goals and all? What areas you further want
I
to
think we
expand?
want to further the biofilm aspect of bacteria is complex as far as the molecular mechanism is concerned. We have not understood everything about it. There are still a lot of novel structures to be solved, lot of interactions to be understood. And I'm quite hopeful that the structural information can then be utilized for drug discovery purposes. So that needs to still be addressed, that needs to be pursued, that needs to be followed. And we hope we can contribute to this in the field.
And also, ma'am, this one is like you have experience in of your postdoc abroad. You are now working for so many years here in India. What exactly can be improved in India when we compared the work culture and the infrastructure abroad?
So I think there is no difference as far as the infrastructure is concerned. There's no difference as far as the questions we are asking. There is no difference in terms of the quality of the science now that is happening in India. There's a lot of support from government for the infrastructure. Students are doing very well. I think a little bit of the support system, the administrative support system can be, I would say, can be improved a little bit, and that would go a long way in supporting scientists. So that's, I think, very important.
And if industries can also involve in funding,
I think that would be wonderful. Yes. That would be wonderful if we can have more partnership with industries, which is happening, I believe. A lot of it is happening already, and that would be also very good for Indian scientific ecosystem.
Then we can translate into
Absolutely.
the research that can benefit society.
Absolutely.
And solve the real world problems.
Yes.
And not just
so RCB has a bioincubator.
Yes.
There are now opportunities for scientists to actually explore a startup. BIRAC gives a lot of support. And now because it's on the same campus, we feel a little easier to approach them and to figure out if there is anything that can be, taken to the next stage of translation. Earlier, scientists would not know whom to approach, where to go, or where to ask questions. So having a bioincubator on campus is very useful.
Yes.
So there is less hesitation in approaching them and to be able to see. So that helps.
Great, ma'am. So, ma'am, what is your is there any piece of wisdom that you would like to leave with our listeners? Something that has been instrumental in your journey?
I think I would be repeating myself if I said curiosity, hard work, perseverance, all of it is required, and passion to be able to do science. And students should continue, persevere, and work hard. That is all I would like to say.
So with this, I think, ma'am, all the very best for your future endeavors.
Thank you so much.
We hope that we again catch up you later.
Absolutely. It would be wonderful.
In future with another topic, maybe with another leads of your research.
Absolutely.
And thank you so much for giving this opportunity.
Thank you, Sonia. It was wonderful interacting with you.
Thank you so much.